Sriji, please start.
Namaskar, a very good morning, friends, and a very warm welcome to our virtual speaker series on Ayurveda. Today's session 24th of season 2, pharmacological and therapeutic effects of top 75 traditional Ayurvedic medicines. And today's topic is the effects of Yogaraj-guggulu. And our expert speakers today, Professor Dr. Praveen Guleria-ji, a very warm welcome, and Dr. Mukund Sabnis, very warm welcome, sir. Thank you for joining. And our expert special guest today, Dr. Jerry Cott, a very warm welcome, sir. We are looking forward to hearing some of the very special comments from you, and our very own dear Dr. Bhaswati Bhattacharya, who is part of this initiative as well. Thank you, Dr. Bhattacharya, for joining. Friends, this initiative, which we started with many other partner organizations and have been running for almost 1.5 years now, the purpose and objective of this promoting Ayurveda as a natural way of taking care of overall well-being. As we know that Ayurveda focuses on balancing your mind, body, and spirit. It doesn't only focus on the symptom, but also it focuses mainly on the root cause of the issues and creating the overall well-being for all Canadians as a preventative tool as well as disease management tool. And our speakers who come from time to time, they spared their time, and we sincerely thank them for sharing their knowledge and wisdom with us. And this is our bi-weekly rituals to share this knowledge and wisdom to all and spread that happiness of well-being. With that note, we're looking forward to the great presentation today, and over to you, Dr. Verma.
Sriji, thank you very much. Good morning, friends in Canada and US, and good afternoon in UK and Europe, and good evening in India and Sri Lanka. Welcome to 24th session of virtual lecture series on pharmacological and therapeutic effects of top 75 traditional Ayurvedic medicines. Friends, we are running this virtual lecture series from Canada with the support of Canada-India Foundation, Toronto, Consulate General of India in Toronto, American Association of Ayurvedic Professionals, USA, Ayurveda Union of Midwest, Chicago, USA, European Ayurveda Association, Germany, Association of Ayurveda Academy, UK, International Ayurveda League, India, Tatha Astu, USA, and our media partners in Europe, UK, Canada, and USA. Today, the topic is pharmacological and therapeutic effects of Yogaraj-guggulu. Yogaraj-guggulu is a traditional Ayurvedic medicine which is prepared from 28 herbal ingredients. Yogaraj-guggulu is used by traditional Vedas for curing rheumatoid arthritis. It's an anti-disease modifying, anti-rheumatic Ayurvedic drug. And it is also used in sciatica, gout, osteoarthritis, even in amenorrhea, obesity, painful periods, and also in hemorrhoids. It is also useful in tinnitus, which is very difficult problem for the modern doctors to cure. Today, we have two expert speakers. We have with us Dr. Praveen Guleria-ji. She is currently working as a professor in Department of Ras Shastra and Bhaishya Kalpana at Ayurvedic Medical College, Sraba, Ludhiana, in Punjab, in India. She has special interest in spreading knowledge of how basic principles of Ayurveda, diets, and Bhaishya Kalpana are applied to achieve longevity. Welcome, Dr. Guleria-ji. And friends, we have with us Dr. Mukund Sabnis, who is currently working on obesity and hyperlipidemic disorders. He has treated more than 50,000 patients of obesity with antioxidant sesamol, which is dried from sesame oil, and hypolipidemic medicines of herbal origin. He has developed SOPs for treating PCOS and coronary artery disease, and he formulated various medicines for these disorders as per individual need. Welcome, Dr. Mukundji. Friends, today we have two special guests. We have with us Dr. Jerry Cott, who is PhD and senior neuropsychopharmacologist and natural product specialist. He retired from NIH, TDR, FDA, USA. Today, we will learn from him how 5,000 years old natural products mentioned in Ayurveda can be brought in Western market. Welcome, Dr. Jerry Cott. And friends, we have with us Dr. Bhaswati Bhattacharya ji from New York. With us, she is MD in modern medicine and PhD in Ayurveda from BHU. She is a guiding force of this lecture series. Today, we will learn from Dr. Bhaswati ji how can we integrate best of modern medicine and best of Ayurveda to help those people who are suffering from those diseases which are considered to be incurable in modern medicine. Welcome, Dr. Bhaswati ji. And friends, we have Dr. Madan Thangaveluji with us from Cambridge, UK. He's a genome scientist. Dr. Madan Thangaveluji will moderate the technical session of today's lecture. So without wasting any time further, I would like to invite Dr. Madan Thangaveluji. Over to you, sir.
Harishi, namaskar. Thank you for bringing all of us together again on the 24th session. Welcome both Dr. Sabnis and Guleria ji, and a special welcome to Dr. Bhaswati. Thank you for bringing Jerry along with you. Now, I have the unenvious task of keeping this to schedule. So we have just 90 minutes. And in that 90 minutes, we have a lot of information to get across. And let me see how to do this. So I've done the first thing of starting a timer here. And starting the timer also alongside putting my screen on. I want to share, before we get on to comments from Jerry and Bhaswati ji, I just want to highlight, just flash a few screens through. And you can see here the plant that we are going to talk about. It's here on the right-hand side. A very precious plant. It's been known for thousands of years and is used across the world in different traditions. You can see the little gum that comes out of these plants. And on the left is boswellia, which is traditionally called frankincense, versus myrrh. While searching through the literature, the best description I found relatively recently comes from the state of Haryana and the city of Ambala in Haryana, from this College of Pharmacy. Talks very clearly about what the plant yields, so many grams, about half a kilogram of dry resin during each collection. It goes on then to give the different chemicals that are found in there. I won't dwell on this, but you can see more details at this PubMed ID. I want to spend a little time here looking at the work of Thomas Burris, who currently is the director of the University of Florida Genetics Institute and professor of pharmacodynamics at the College of Pharmacy. There's Thomas there for you. On the left and right-hand side are the two stereoisomers of perhaps the most important active ingredient that we are going to be discussing, that we learned from Dr. Bhaswati ji and Jerry. Now, also, I just want to flash very quickly the work of Professor Herman Amon from the Department of Pharmacology at the University of Tübingen, showing very clearly how, in this instance, boswellic acid inhibits inflammation, and shows how you can go on to prevent type 1, type 2 diabetes by suppressing the expression of pro-inflammatory cytokines. So there's much more available in the literature about these things. I will leave it here. And I want to start off by inviting Jerry to share his thoughts. Jerry, thank you for joining us today.
Oh, thank you. I guess the best way for me to tell about how I came to know about Ayurveda was when I was starting a natural products program at the National Institute of Mental Health. I became acquainted with chemist Dr. Renu Misra. Many of you probably know her. I mentioned to her one day when we were at lunch that I had high cholesterol and I had started to take a statin drug to lower my cholesterol. She said, "Oh, you should take Guggul." So I said, "Well, what is that?" And she explained about Guggul, and she actually had some that she had brought from India. It was from a Western company, westernized company, I guess, Cipla, and they produced this, and it was in a package, you know, a very nice package that had a very standardized looking product that claimed to have 25 milligrams of Guggul in each tablet. And so I left off my statin drug and started, which wasn't working very well, by the way, wasn't lowering my cholesterol very much. And started taking her Guggul, and within a fairly short time, my cholesterol went way down. And this was my first introduction to an herbal medicine that really had a powerful effect. But I guess up until that time, I thought that herbal medicine was just weak medications, just weak drugs. But this one was obviously a very powerful drug and had a very powerful effect on me. And the interesting thing is that I didn't have enough to last very long. I had enough maybe to last for about 3 months. And then my cholesterol went back up again when I ran out. I had the opportunity to go to India a year or two later and went by a chemist shop and bought several packages of that same product from Cipla, brought it back home, started using it, and it didn't do anything. It didn't work. So we had a friend, Ramesh Pandey, who had a company in New Jersey. I asked him to analyze the product that Renu had brought back, and he said it only had 5 milligrams of Guggul. And Renu and I were speculating about what might have been the problem, and she seemed to feel she had some information about the way that Guggul was supposed to be produced for lowering blood lipids was that it needed to be aged for 5 years before it was put into the formulation. And we speculated that maybe the company got anxious and decided not to wait the full 5 years. We didn't really understand what kind of oxidation process might have been going on to produce more Guggul, but we thought that might be the case, but we don't really know. And I never really found out why the product that I bought didn't work. So I would like to put that back to you experts and see if maybe you know what could have happened. And of course, Cipla is not known for Ayurvedic medicines and probably didn't put it together correctly in the first place, but I'd be very curious to know.
Jerry, thank you. Thank you so much for giving us your personal story on how you came into Ayurveda and how this unfortunate experience. It is a mystery. We will hear a little bit about this from our experts about those purification steps, the different geographic locations from where these things can be obtained. We'll hold on to that thought, but we'll come to it perhaps during the discussion. Baswati-ji, thank you for joining us, and thank you so much for bringing Jerry along with you, and for Jerry to share this wonderful story. Now, the floor is yours, Baswati.
So Harishji has asked me to talk a little bit about how the modern medical doctor will interpret this. And as Jerry has said, his first interface with Google was in a clinical scenario where he was looking for something to replace what didn't work. And that is where many people find their first interface or experience of Ayurveda. There are many things that I learned about Guggulu once I started studying Ayurveda, and I think Dr. Guleria and Dr. Sabnis are going to talk about this. I was sitting at breakfast with Jailan Shastri, who has done a lot of research with Guggulu, and he was saying that you have to challenge any plant or animal against its boundaries in order to get it to express its best self. That challenge is important. So when you see an oleo resin growing in a moist environment, and we know that oil obviously is snigdha or moist, you won't get very good Guggul. But if you grow it in a very dry place like Rajasthan or near a desert where there's just enough water, but a lot of wind, a lot of dryness, a lot of challenge with hot sun, then you'll get very high levels of Guggul. This is a very logical thing to think about. And what modern medical doctors need is a mechanism of action that takes that way that Guggul will grow from a botany that they will generally understand over into how does it work clinically? How would that oleo resin come into a cell and perhaps go in and create an environment, a local microenvironment where that cholesterol that's stuck to a wall will be able to be lifted off. So Jerry was very successful with his Triphala Guggulu clinically, and I think that will attract modern doctors. But the mechanism of action has to be given in a logical way. So I ask my medical students, how do you remove that dried oil on the outside of an oil bottle that's sitting near the stove? The stove has made it very hot and dry. How do you remove it? And they will say, well, water. I say, water doesn't remove it, right? They say, yeah, it doesn't. Scrape it. I said, well, then scraping will destroy the label on the bottle, right? I said, yeah. How do you remove it? It's to put more oil on it. And what oleo resin does is it goes in and interacts with that, shall we say that, in English, we'll say the yucky oil, we call it Vikriti in Ayurveda, and it will remove that. And so Guggul has been used in actually about 200, 300 different combinations that are commonly used and thousands in the ancient textbooks for removing that resin that we call ama, or some call it toxin, but it's really a residue that's sticky. And so I'm looking forward to hearing from Dr. Sabnis and Dr. Guleria in a very common way for doctors logically to understand, and maybe some molecular structure to understand how that oil will take sticky oil and carry it away so that people find relief. And in fact, we find clinically that fatty liver can be reversed, pancreatic problems can be reversed, atherosclerotic plaques can be alleviated. And these are very strong assertions, because people think that herbal medicines have no medicinal benefits because that's what the NIH has set up through the NCCIH. But the reality is that herbal medicines have actual very strong medicinal qualities, and that's been shown in humans. Maybe not in America, because in America you can't show dietary supplements as being medicinal, but in India, where it's a legal medicinal system, Ayurveda has been shown to be very useful in humans. And I look forward to hearing from our speakers about the possible mechanisms so that modern medical doctors can understand and love and appreciate Guggulu more. Thank you.
Baswati, thank you so much for sharing your thoughts. Now, we still have a few more minutes before we go to our expert speakers. So I pose this question both to Jerry and yourself, Bast. What if Jerry did not get that initial effect on the first batch that he tried. We wouldn't have him here talking about it, would we?
We probably would, because Jerry is a natural products expert, regardless of his personal experience. But Jerry, you have more theory. Jerry, go ahead.
Well, that's a good question. I'd already started a natural products program before then, so my interest was definitely there. My interest in Ayurveda specifically might have been retarded a little bit if I hadn't been for that experience, though.
Jerry, what is important here? I flashed one of the figures where we talk about, in this case, the Guggul sterons, and we talk about the E-type and the Z-type isomers. And you made a comment about aging, and we will hear from our experts about the purification steps. They call it Shodhana, the purification steps that they take this through. Now, a lot happens over this process. Keep in mind, what we've seen, what we see today in the best of preparations is a kind of coming together of thousands of years of what would be some form of chemistry, attempts at purifying things, attempts at understanding how they react, etc., which a modern chemist will do differently. And we will hear about those two, the purification process in particular. Spontaneous racemization is known for a lot of chemicals that you age things and it spontaneously racemizes. We don't know what the mechanism is, but clearly these are the kind of questions that open, perhaps when we come into doing finer research. These are the kind of things where we might be able to bring together an interaction between the Orient and the Occident, and to put it as crudely and bluntly as that, and perhaps more specifically in terms of a research program between India and America and Canada. I don't know if you wanted to just address this before we go to our two expert speakers, both Baswati, yourself, and Jerry, perhaps starting with Jerry. Do you see something in here to generate that vibrant research program for the future that brings together the best of the East and the best of the West?
Well, I had the bounty of visiting NIMHANS in Bangalore when I was with the NIMH. It was a joint meeting between the Indian National Institute of Mental Health and Neuroscience and the American NIH. And we were looking at treatments for mental health disorders, since that was the institute I was with. And we were comparing treatment of primarily schizophrenic patients who were diagnosed and treated by Ayurvedic physicians versus Western medicine physicians from India. And they showed the interviews with these patients and with translations for the Ayurvedic. And everyone there was fascinated by this kind of comparison, this head-to-head, not to see which was better necessarily, but just look at the approach, the different approaches that the two sides were taking to look at the same problem. And I think that doing this in any other illness, diabetes or whatever major illness that of interest, if you can approach it like this and have scientists on both sides explain their approach and look at the results were good in both cases. I think this really will help to get people involved and bring them along. Yeah.
Thank you, Jerry. Mental health, of course, is a very important theme in the India-Canada interactions. Perhaps you know, we had the Assistant Minister of Mental Health and De-Addiction recently in India, Minister Tibolo. And there are ongoing discussions with Canada. Perhaps there's an opportunity here to also draw in, based on your experience, to draw in United States also into these discussions. We will leave it there for the moment. Bas, would you like to comment on this as we have a few more minutes before we move on to our two speakers. Thank you.
I definitely think that there's a potential for research programs, but I think currently it's not being done in the way that the bureaucratic overview has intended, and that is to have a transdisciplinary, interdisciplinary, multidisciplinary program. So it doesn't just mean having biochemists sitting next to organic chemists. It means having people from all different walks of life, having a bio-inorganic chemist who can talk about why bhasmas work so well with Guggulu, as well as having a person that is a botanist who can talk about oleo resins. You mentioned frankincense, myrrh, which is known in the West because the three wise men that came to visit Christ had frankincense, myrrh, and gold, right? So there's the gold, the metal. Why were those the three wise men? Has anyone ever discussed what the chemical, biochemical reasoning is of giving a newborn child frankincense, myrrh, and gold? I think that's worth a discussion. But you're only going to have that discussion when you have people of all different disciplines. So in this case, I would love to have someone talk about the L and R, the E and Z, the stereoisomers, right? The racemic relationships, and how one happens to occur more biologically, and that is inspiring the immune system to work well, whereas the exact same molecule in its stereoisomer is seen as an enemy and oftentimes incurs the wrath of the immune system and creates inflammatory cascades. Now, in the diagram that you showed that I could just see, when you add an icosanoid, which is a 20 carbon molecule, to the mix, there is a new kind of molecular biochemical reaction that can happen. Yeah, so there's the stereoisomer, and there's those 4 rings. And if you count the carbons in the way the chemists do, you'll see that these are very similar to the icosanoid series, right? And then you went to, I think, the next slide where you showed that there were pro-inflammatory markers that, there you go. You see how on the right side, you're showing how the boswellic acids have an inhibitory action on the inflammatory cytokines. And those are all derivatives of the arachidonic acid cycle. So on a molecular basis, I hope Mukunji will talk about this, you have a chemistry side of things, which many different people in the chemistry world, biochemists or inorganic chemists and various pharmacologists and toxicologists would like to see. But then that needs to be translated for the people that don't understand that language. So the botanist or the historian or the people that understand Guggulu in this case, from all different walks of life, if they could be invited around the table, no one will have all the answers, but together, when you have an interdisciplinary group, pieces will jog the understanding of different experts. And that's what creates these solutions that so far have been unavailable to us, and a deeper understanding. Because ultimately, as a pharmacologist and as a clinician, a physician, I really want these solutions for my patients who are really suffering with problems where they cannot find a drug that works for them. They'll use anything, but they cannot find a drug. And frankly speaking, one of the reasons that I'm in Ayurveda is that, and took the time to do the PhD in Rasa Shastra, is that there are cures known in Ayurveda, if it can be communicated correctly, that absolutely reverse diseases that we do not have a clue about in modern medicine, whether that's modern medicine in America or Canada or Europe or in India by the MBBS doctors. And many times they come to Dr. Subnis and ask him, to collaborate because he understands how to communicate that information. So in summary, yes, I think a research program is possible, but whoever is the head of that commission or group needs to have the open-mindedness to involve all different kinds of people and not just what their brain thinks is the usual candidates. I think that's some barriers. Yes, thank you.
Thank you, Bash. Stay with us, Jerry and Baswati, please stay with us. Let's listen to our experts. And then I'll try and rush them through. These are only taste tests. The whole idea is to get the discussion going. And I'm delighted we've got that discussion going here. And there's much more to happen. Jerry, particularly with the mental health area, we know there's inflammation associated with many aspects of mental health. And we got to understand these things. Perhaps this discussion, what we've started here, we can try and extend over the next months and years. But before we continue, thanks to both of you for these early comments. And let's proceed. Mukundji, namaskar. Thank you for being here with us. You've heard some sharp and sometimes serious comments, quality, diversity. Are we explaining what we know in Ayurveda correctly, the long history, etc.? Let's hear from you, sir. Thank you for joining us today. I know that you are one of those key people working with Guggul and Google Steerons. Let's hear from you. Thank you for joining us today. If you're sharing a presentation, the floor is yours. I hope you can. Jerry, while you listen to Jerry Bash, you listen to the two speakers, and the question that will come out at the end of this, I hope, and I hope we'll have time for it, would be which way forward? Thank you, Mukundji. If you could put this on presentation mode, then thank you. All yours.
So, greetings to all of you. And
Mukundji, there is a little bit of a background noise on your.
yeah, now is it there? Now is it there?
Yes, there's a ticking going on. It might be a clock, a very loud clock in your.
No,
but doesn't.
Is it now?
Let's continue. We won't worry about it. But apologies to the viewers for that.
Thanks to give me a chance to share my views among you. And today I'm going to talk a little bit about the pharmacodynamics, the analytical part and analytical chemistry of Google. And I'm more interested to explore its mode of action for the practitioners. Along with that, Google is a very versatile drug and it interacts differently with modern medicines also. So that is also to be known to the practitioners, basically because Google is taken for a very longer duration and concurrently it is taken with maybe antihypertensive, maybe thyroid medicines and maybe much more medicines working on metabolism. So what are the exact interactions and how we have to tackle with it? That is also a very important subject when we talk about Google. Google has been mentioned since Vedas and there are quotations in Atharva Veda also regarding Google and they have clearly mentioned that Google is used in Yakshma. Here the word Yakshma is moreover related with tuberculosis, but this is not a tuberculosis, it is a group of disease. Yakshma here means it is a group of disease where Guggulu can be used, has been explained in Atharva Veda also. So directly going to the formulation of Yograj-guggulu and then coming to the Guggul preparation, that everybody knows that it is a Comifera mucul or Comifera vegetae. And here we have seen that there are 28 type of formulation ingredients in Yogaraj-guggulu. Coming to the properties of Guggulu, which is used in Yogaraj-guggulu, we have to talk about that. When we go through PubMed or when we go through the research papers, what we have seen, one thing we are missing very badly, that the research papers have not mentioned that the Guggul which was used was Nava Guggul or Purana Guggul, because the properties of new Guggul and Purana Guggul are exactly opposite. So what type of Guggul, what is the age of that Guggul? That is very, very important when we are using Guggul in the for some ailments. Second most important thing, there is one process of using Guggul and that process is called as a Shodhana, that is a purification. Now when we are extracting Guggul, because when we talk about Guggulusomes or when we talk about Guggulusterols or when we talk about Guggulusteroids or Guggulustron E or I-Guggulustrons, so when we talk about it, those are completely directly extracted by the solvents. No Shodhan process is undergoing all these things. And here Shodhan of Google is very, very important. Shodhan here is not only for removing the waste dust materials and some unwanted materials, but it also has some chemical changes. It also has some pharmacological changes. And this Shodhan process, whether the Shodhan has been done by Triphala or whether the Shodhan has been done by the Shamulakvat or whether the Shodhan has been done simply in water, that has never been mentioned. That is the most important thing. Third most important thing what I want to elaborate, and which is the highest important thing, is about the dosage of Guggulu used in the treatments. Dosage of Guggul plays a very, very important role, because the dosage which has been mentioned in Sahitas and the dosage which are been mentioned in the what we can call as the granthas which are written after Sharandhar Samhita, there is a lot of difference. Fourth thing, why people are not getting good results, what has been promised in Sahitas. Most important thing, Sahitas, that is Charak, Shishrut and Vagbhat, they have not advised or they have not used Guggul in the form of vati or in the form of tablets. They have never used. I'm going to show the quotation. These are very important things. And they have used in the form of either they have mixed it with mouncerus, or maybe they have mixed with some juices, or they have some mixed with some decoction, and they have used it in the liquid form. They have never used it in the vati form or in the tablet form, because the dosage they have mentioned are much higher than the dosages we are using in today's world, that is 250 milligram or 500 milligram in the form of BD or TDS, maybe anything depending on the physician and depending on the disease status. So these are the four things: either what the Shodhan, the age of the Guggul, the dosage which are used. So these are the things, and the temperature of the processing of the Guggul when it is getting Shodhan. Because the flavonoids, the flavones, the steroids, they are heat liable. At what temperature you are doing Shodhan, that is the most important thing. And we have found that when the Shodhan is done below 40 degrees centigrade, then it retains maximum amount of steroids and sterols, which are the active ingredients in Guggul. But majority of the time, when the Shodhan is done, that's boiled, and the sterols and steroids, they disintegrate or they get oxidized, and then we don't get a very good amount of results. Now, if you can see in this sutra, that is Bhagna Sandhana Krid Vrishyaha Sukshmaha. So what do you mean by that? That Guggul is used to join the broken bones. Bhagna Sandhana, it is useful in fracture, but here Nava Guggul is only useful in fractures. Purana Guggul can't be useful in fractures. So whatever are the then Medo Mehescha Amuvatascha, clay the Kushtama Marutan. So these Meda, that is obesity, Nava Guggul will never can't be useful. Here Purana Guggul will only be useful when we. So that's why the age of the Guggul is very important, and they have clearly mentioned Sa Navo Brahano Vrishya Puranasto Atilekana. So whatever the Nava Guggul is there, it is useful in the catabolic disorders. It is useful in catabolic disorders, and Purana Guggul, it is useful in anabolic disorders. That is the clear classification for the general practitioners. We'll see the chemical structures and what changes are there in the Nava Guggul, and that is a different story. But when we come to the results, we have to know that which type of Guggul we are using, and that's the most important part. So there are a lot of studies which has been undertaken for the in vivo studies are there, in vitro studies are there, then studies in different type of diseases like asthma, fibroids. So lot of studies are there, lots of research papers are there regarding Guggul, but none of the paper haven't thrown any light on the age of the Guggul, the dosage which has been mentioned in Samhita, the form of the Guggul, and the temperature of the processing of the Guggul. These are the four important parts when Guggul is to be explained. So when we talk about the pharmacology of the herbs, here when it comes to the herbal pharmacology, we in Ayurveda we explain the pharmacology in the terms of rasa virya vipak. That what is a rasa? That is maybe in Purana Guggul it is Tikta ras, Nava Guggul it is a Madhur ras. So vipak is Katu and the virya is Shita or virya is Ushna depending on the age of the Guggul. But nowadays we are able to explain the pharmacological impacts to the extra gustatory taste receptors, maybe T2R1, T2R2, or maybe other sweet taste, bitter taste, or astringent taste, umami taste receptors. Depending on the stimulation of these receptors, we can explore the pharmacology. And then third, we explore the pharmacology as per the secondary metabolites, maybe steroids, phenolic compounds. So all these things we can explain it very nicely. So I'm trying to explain it in the terms of all the three parts here. So these are some of the chemical structures of Guggul sterols, which 3 to 4 days back I have analyzed in my lab. That what are these chemical structures and how they are to be identified. Exactly because we are not using Guggul as a single. See, Ayurveda has never used Guggul as a single drug. Guggul has every time been combined with majority of the different type of herbs or different type of metals. In Yogaraj-guggulu, Sharandhar explains there is a different quotation. They are just given an optional that there are certain metals included into it. Or Guggul or some other drugs are also introduced, maybe Gokshiradi, maybe Maha Yograj, maybe Kanchanar. So different type of herbs are introduced into Guggul, and then Guggul is used. It is never advised to use Guggul in a single way. Either it is used with Shilajit also. So these Guggul sterols, these Guggul stron E, this Guggul stron Z, these are isolated phytochemicals which Ayurveda doesn't expect. Ayurveda doesn't. So okay, for the exploring the pharmacological impacts, we can take the help of these. This is not a holistic approach. So if we want to explore the pharmacology, these phytochemicals come to help to us. If we want to go for standardization, this phytochemical comes for the help. If we want to explore the molecular biologic patterns, we can take the help of these phytochemicals, but we can't use these phytochemicals as it is in our formulation, neither we use it. We use it in the form of different type of formulations like Yogaraj-guggulu, what we are going to discuss today. So these are the certain quotations which are coming from Charak, which are coming from Sushruta, which are coming from Vagbhat, and these quotations clearly explain that Guggul is never used in the form of tablets. It is to be used in the form of mixing with the decoction, or mixing with certain juices, or mixing with some chicken soup or mouse ras and all these things. We have to mix it with, and then we have to take it. So these are certain very important quotations that Guggul is consumed by mixing it with decoctions by either Brihat Panchamul or Laghu Panchamul. In the disease of Vata Kapha can be consumed by mixing it with warm water. Then so certain different type of things are there where we can mix Guggul. So in Vata Shanita, maybe in gout, Guggul is consumed by mixing it with cow's milk. And the same way Shilajit is used. So Shilajit and Guggul, they work parallelly, they work together, and they have advised it that these two things are to be mixed together. So Rasena Payasa Mutravata Pitta Kapha. So Guggul is consumed by mixing it with meat juice, cow milk, cow urine, respectively in disease of Vata, Pitta and Kapha. So whatever the vehicle we are using along with that is called as Anupan. That is also a very important, and these interactions are very important to evoke a pharmacological response. Dose is also very important. I'm going to discuss about the doses. Dose is also important. So Guggul is consumed by mixing it with different liquid juices and decoctions. Sanhitas, Ms. Charak, Sushruta, Vagbhat have never advised you to take them in the form of tablets or pills. So coming to the Yogaraj-guggulu, which has a 28 type of ingredients into it. Main ingredient, no doubt, 27th part is Guggul, and that Guggul we are going to discuss with. And this is just 3 days back I have analyzed the Guggul in my lab, and this is very interesting that 3 companies which are leading companies in India, I have decoded it, which are leading company in India. These 3 companies, these are the FTIR fingerprint of these 3 companies, and here we can clearly see the peaks at 1000 in all the graphs, and these are the peaks of steroids. That is maybe Guggul stron Z, Guggul stron E, and when we go to the 3400 or 3266, there are again similar peaks coming to it. These peaks tell about the sterols in Guggul, and when these are intact, means if we have processed this in a very perfect way below the temperature of 40, when we are doing Google Shodhana, these Google scrolls, the peak might have shown a higher intensity. These might have shown the higher intensity, and then the peaks what you can see between 1500 here, or the peaks you can see between below 1000. I have given the chart that these are some CH stretchings are there, CS stretchings are there, then there are some bendings in the carbon-carbon double bond or single bond. These are all processing effect on the Google when we are doing Shodhan on it. If the Shodhan, whether the Shodhan is getting correct under the proper temperature for a proper duration, we can easily analyze it with this type of fingerprints, and then we can tell, yes, the Google which has been prepared is in a very, very correct way. So there are certain, here you can see in this graph that there is many more peaks between 1000 and 500. These 1000 to 500 peaks, they show about the aromatic compounds, aliphatic compounds, so in the phytochemicals into the shunta, miraj, or maybe in ginger or pepper, or all some drugs, herbal drugs which has been added to it. So there are carboxyl groups over there, there are alkenyl groups over there, there are amine groups over there, which are coming from phytochemicals, and those we can clearly identify with these fingerprints very nicely. Here there is a different, again, analytical part with the Google, that is what we can, so that the metals coming into the Google, and here whatever the formulations we have analyzed, those formulations were devoid of metals. Still this much amount of impurities are coming into Google. Aluminum is not expected, silicates are not expected. Still these aluminum, these silicates are present over there in a very good proportion. That shows that these Google processings, they have lot of impurities into that. So if the Google is not processed very correctly with a very high temperature, which will not have a good Google sterols and Google steroids, and if such type of impurities are there, and if they are given in not in a pharmacological dose, whatever people are using Google, they are not using in the pharmacological dose. And if you are not using Google in a pharmacological dose, that to a complete Google, that's a formulation, no doubt of getting results. You'll not get results out of it. So here again a different problem is there, that again we analyze the same Google of the three companies, and if you can see the 3 number disintegration, none of the tablets exhibited disintegration within the specified time frame of 15 minutes. If that medicines are not getting disintegrated within 15-20 minutes, then what's the use of doing the tablets out of Google? Because they are never completely disintegrated. The pressure we are applying while doing the press in the press machine or in the tableting machine, that's too high to make it a tablet, and then the Google don't disintegrate completely, passes as it is, and then we can't get the complete results out of Google. These are some of the important the dosage form what we Indians are using Google. Then coming to the Yogaraj-guggulu formulation, here there is one option. Sharandhar has clearly mentioned Vangam Ropyam Cha Nagam Cha Lohasarthabrakam Manduram Rasasundaram Pratyekam Palasanmitam. I have highlighted it because they have given this into the bracket, and when this is added, it is called as Mahayogaraj-guggulu. When these metals are not added, it is a simple Yogaraj-guggulu. Then how this Google works? Now see, Guggular Yogarajoyam Tridokshagna Rasayanaha. So when Google is used as Rasayan, it is a complete, can you imagine, they have mentioned a dose of 4.8 kg in a cumulative dose when we want to go for Rasayan. How many of us we are using? We don't use this type of doses. So when you want to, as a Tridoshagna, or when you want to use it in Rasayan, they have given a concept. See, the concept of cumulative dosing is since Ayurveda. They have given that 4.8 kg Google has to be consumed in a specified time period. This is a cumulative dose. If you want a Rasayan activity. If we want to Maithuna Ahar Panaram Tyago Naivatra Vidyate, here it is a very, very clearly specified that when you are using Google in such a dose, there is no need of any dietary measures. So potent is Google, but the dose that is also to be used in a pharmacological dose and for a specific diseases. Then Sarvon Vatam Amayan Kushthanshi Grahani Gadam Prameham Vataraktam Cha Nabhi Shulam Bhagandaram. So it is used in diabetes, it is used in obesity, it is used in polycystic ovarian diseases, it is used in infertility. So reto dosha haram pusam, rajo dosha haram striyam. So reto dosha, there are many obese people who have low testosterone due to the low nitric oxide synthesis into the penile arteries. Here Google can work very nicely, but it should be a Purana Google. In PCOS, Rajo Doshahara, it is a PCOS where it can be having an impact on insulin sensitization at an ovarian level. How it is going to, I am going to show it. So these are certain impacts where we have to use Purana Google, where we have to use Nava Google. That is to be very clearly identified first, and then we should use it in the patients. Then they have used Anupan, that is a vehicle, and for different diseases, different Anupan have been mentioned. That I have highlighted into red color, that for different disease, different Anupan has to be used. So Google is not singly used. If somebody is not getting results, it is to be used along with Anupan. It is to be used in a proper pharmacological doses. Then and then only it will give a proper results, or otherwise it will not elect a pharmacological activity. So Google controls anabolic in Santarpanjani disorders. That is, it is a Nava Google. Nava Google is contraindicated in catabolic disorders. It's very clear. It is advised. Purana Google is advised only in Santarpanjani disorders. Nava Google is advised only in Apadarpanjanya disorders or catabolic disorders. So this is the way how we have to first classify the use of Google, where we are using it. On that it depends which type of Google we have to select for medicine preparation. Unfortunately, none of the companies which are preparing Google in India are mentioning there is no label claim whether the Google which is used is a Purana Google or Nava Google. That is the biggest problem. So these are the Anupan, this is the adjuvants along with Google. That is a big list over here. I'll skip it. Now coming to the Google sterols and Google lipids and Z-Google-sterone and all these things. First of all, those are all insoluble in gums which are soluble in insoluble in ethyl acetate and insoluble in ethyl acetate. So again, these are all soluble into the solvents, and we people are using water-soluble extractives. We never use maybe ether or ethyl acetate or alcohol or chloroform extracts for the preparations. Where modern medicine, they use different type of solvents to isolate the phytochemicals, which is not at all expected by Ayurvedic practitioners, not at all expected by Ayurveda. We only use water-soluble extractives. Only water-soluble when it comes to Guggulu, or we may use hydroalcohol extractives. Only such type of extractives we are using. So Guggul lipid is not an Ayurveda preparation as per my opinion. If we are comparing Guggul lipid and if it is reducing cholesterol, the mode of action means when they talk about Dhamani Praticha, it is not directly because the biomarkers which are used for Ayurveda research should be totally different from the biomarkers which are used in modern medicine. That's very important. If I am looking that okay, cholesterol is getting reduced, then the mode of action is not like HMG CoA reductase activity. There are different type of activities which may be having impact, and what are these different type of activities we are going to see. So the basis of the cholesterol lowering activity of Guggul-sterone is the inhibition of FXR activation. That's very important. This FXR activation is the most important thing when we talk about impact on the metabolism, impact on cholesterol or cardiovascular disease or atherosclerosis.
Mukunji, I'm going to interrupt you here because could we come back to this? And I want to bring Professor Guleria into the discussion.
So just 2 minutes, sir. 2 minutes. Just I want to talk about interactions. 2 minutes.
Please.
Yeah, I'll finish it. Basically, this Guggul is having a anti-inflammatory activity, which is very important. What you mentioned about the interleukins and cytokines and all these things. It has an activity on inflammation. It reduces inflammation. It works through bitter taste testants and receptors. Then it is having a potentiation of glucose-stimulated insulin secretion. So these are it works on the gut hormones. So these are the different mode of actions here. And some of the large slides that Guggul interacts very badly with aspirin, warfarin, then potentiates the effect of aspirin and warfarin. Then it has a stimulate the effect of thyroid gland. These are again the interactions which it should not be used consequently. along with the antithyroid drugs, and it should not be used concurrently with the beta blockers because it reduces the uptake of beta blockers from the gut. So these are some of the important untoward effects, and the doses which are given, they are on a very higher side. Those should be focused very correctly. We can go to the text and we can again discuss about the doses. Thank you very much for the time which has been given. Thank you very much.
Thank you for the sake of time, but your very rich pointers to where future research might go. Harishji, please, before we go to.
Yes, Dr. Mukherjee, one question only. If the Yogaraj-guggulu in a formulation form is available in the market, say 5 years old, can it be considered as a aged Guggul?
No. When we are using it, when we are processing it, at that time it should be 1 year old. When we are processing it, when we are already processed, then it is not to be considered as an old Guggul.
Thank you, sir.
Mukherjee, before we go to, perhaps this point will be clarified by Professor Guleria. Thank you, sir. Please stay with us while we listen to this point about aging and aspects, the fine details of how we show in our procedures. Dr. Mukund, could you, if you stop sharing your screen, then I think Praveen will be able to join us. Praveenji, namaskar. Thank you so much for joining us from the lovely state of Punjab with its own tradition of Ayurveda and the Punjab Parampara. We look forward to learning from you. There are several questions that have come out here about how we identify quality of products in the market. That's the comment that Jerry started off with. But let's listen to what you have to say, Praveenji. Now, apologies again for if I have to interrupt you somewhere as we progress.
Okay.
So wherever you feel you can speed up and flash through a few more slides swiftly, then we do that. Thank you.
Thank you, sir. Okay, a very good evening to everyone. So Yogaraj-guggulu, the pharmaceutical aspect. First of all, I would like to thank Dr. Harish and all the panelists over here for giving me the opportunity to present my views on Yogaraj-guggulu and the processing of Guggulu. It was a delight to hear Dr. Mukund talking, and unfortunately, because of the time, we are limited. So I'll just share what I have to say. So here I have taken the pharmaceutical aspect of Guggulu, and here's the title and content layout. I have discussed Guggulu Kalpana, discussed how it keeps the diseases away, how it developed as an independent dosage form, what are the key features, the ingredients, method of preparation, and a little slight insight into the pharmacological aspect, which Dr. Sabnis has already described. Then Yogaraj-guggulu will be discussing the ingredients, the method of preparation, and then we'll talk about the standardization. Then the concept of Guggulusomes, which is developed in the coming up in the new pharmacological studies. Then indications, dose and sevan vidhi, the probable mode of action, which Dr. Sabnis has already discussed on the phytoconstituent basis, and then we'll conclude the. Now, this is the Guggulu, the name, one that keeps the diseases away. This is a sample of Guggulu, which we received in our for our personal use in the pharmacy. So, and as already shared by Dr. Sabnis also and Dr. Madan also, this is the Commiphora mucul. This is a plant which is in South India in a herbal garden. We can see the leaves and the fruit is present over here. It is widely used in Sahitas where it was dissolved and filtered in suitable decoctions used orally or locally. So the Dr. Sabnis rightly said the preparation of Vati Kalpana was popularized by Sharangdhar Sahita in the Vata Kalpana in Madhyam Khand. The various dosage forms where Guggulu is an ingredient, these are Kwat, Rasakriya or Avle. That is what we find in the ancient texts. For local applications, we have it in the form of lep, that is a thin application, Pradeh and Malhar formulations. Then we have it in certain grit and tail applications. Then it is used in certain churn and for dhupan, fumigation purposes it was being used. And then in certain ras yogas as Vati or as
modak it has been used. Now, how Guggulu developed as an independent, I'm sorry. How Guggulu developed and why Guggulu developed as an independent drug dosage form. The reason lies in its pharmacological properties, which Ashtang Sangrah Uttar Tantra, Uttar Sthan has rightly described its property as being Yogvahi. Guggulu can be used in almost all the preparations, all the diseases, depending on the compound drugs with which it is being formulated, because it enhances the properties of other drugs without losing its own. So that is the key feature with why Guggulu is being used in a wide array of diseases. Now, Guggulu as Vati Kalpana was formulated by Sharangdar Samhita in Madhyam Khand, where he said, Lehavat sadhyate vahno gudo va sharkara tatha Gugglur va kshipet tatra churnam tan nirmita vati. And so it can be used without use of fire, also by pounding method tablets can be prepared, or with the use of fire as a Agni Pak Vidhi, Guggulu Pak can be done, and then quantity of additives as fine powder should be the same as the amount of Guggulu. Then there is again a variation in the amount of powders we are using and in the amount of Guggulu, depending on whether we want Guggulu to be the main acting agent or it is just a Yogvahi effect we are having. Now, Guggulu Kalpana, what are the key advantages or the key features? Guggulu being a binding agent in itself, no binder is required. The Yogvahi property of Guggulu helps in potentiating the properties of the herbs or minerals it is combined with in a formulation. Rasayan potential of Guggulu with Sar, that is the mobile property of Guggulu, Vishad, that is clarity, and Sukshma, fineness properties, they play a key role in delivering the medicines to their target organs. The action formulations are further potentiated by Anupan, as already discussed by Dr. Sabnis. The shelf life is better than the herbal dosage forms. If we see the shelf life notification of Government of India, the Guggulu Kalpana, it has a shelf life of 5 years. It is considered to be potent for 5 years since the date of manufacturing. The only disadvantage with traditional Guggulu preparation, which Dr. Sabnis already discussed, is the tablet hardness and the disintegration time, which is a major deterrent factor in the Guggulu formulation as a Vati Kalpana. Now, I'll discuss the basic formulation, what is being prepared today, what is available in the market. It has 3 main ingredients. One is the Shuddha Guggulu. Guggulu is, Shodhan is done. And then certain herbs, minerals, or other drugs are used as prakshep, churn, in a powder form. Equal, the amount of Guggulu and the amount of powder, if we are making a typical Guggulu formulation, that has to be equal. And then Grit or Arand Tel in quantity sufficient, enough quantity so that the Rukshita of Guggulu is reduced. So this is the basic formulation. Then we go on to the method of preparation, how Guggulu should be prepared. Every Guggulu Kalpana has to be formulated after Guggulu Shodhan. What are the classical methods of preparation we are going to see? And Guggulu Pak, Prakshep addition is done, Grit addition is done, and then it is rolled into Vati, dried, packed, and stored. Now, Guggulu Shodhan, we have discussed 2 criteria, the method and the medium of Shodhan. The Shodhan methods, which are described in ancient texts in different contexts, dissolution and filtration. The Guggulu is dissolved in herbal decoctions or specified liquid media by heating and filtering, and on complete dissolution, it is dried in intense sunlight. That is the method we find in Harit Samhita and in various ancient Samhitas. Swedan or the Dola Yantra method, here the Guggulu is placed in a cotton cloth. We tie it in a loose bundle. It is boiled in a liquid medium. In a Dola Yantra, we call it, and until maximum Guggulu enters the liquid from the cloth. Now, that is what the concept is. We get the water-soluble principles. Somehow, the decoctions also add on to the extraction of the required phytoconstituents from the Guggulu. After cooling, the sediment is collected and used as Shuddh Guggulu. Swedan and Grit Bharjan is another method of Guggulu Shodhan, where small pieces of Guggulu, they are fried in ghee, placed in Dola Yantra. After the frying process, they are placed in Dola Yantra in specified liquids. And then again, in Sharangdhar Tika, we get another reference where Guggulu is immersed in ghee for 7 nights and taken out and then used. So these are the various Shodhan methods which are being followed. Then The decoctions. There's a wide array of herbs which are being described for the Shodhan of Guggulu, and then they are more specific. Like if you want to use it for a particular disease, like in Sthalya Chikitsa, like in hyperlipidemic patients, the Gomutra method is considered better than the other methods. If you want to have the Vathar action, then the Shmul Kwath is a better option for adopting as a Shodhan medium. Then these are the various drugs which are being used for the Shodhan. Then what are the classical methods of Guggulu preparation when we come to the Vati preparation? If we talk about what are the classical methods, we have the reference of Sompak, or where there is no use of heat. It's the Kuttan, Mardan or Bhavana Vidhi. This is in context with Rifla Guggulu. We give 7 Bhavana of the liquid media are given to the Guggulu. And 1 Bhavana we consider complete when the first liquid it completely dries off in the Guggulu. Then the second method is Aditya Pak, where Guggulu is mixed with other powders. And after that, Dashmul Kwath or any other herbal decoction is added, and drying is done in sun rays. Addition of Kwath and drying is repeated for 7 times. That is the Aditya Pak method of Guggulu preparation. Then Anal Pak, the most commonly formulated formulation is Anal Pak. In this, Guggulu is melted in liquid media. Heating is done till we get the Pak Lakshan. Other powders are mixed in it, and then it is rolled into a pill or a vati. Then these pills are then kept in glass or porcelain jars, free from moisture, stored in a cool place. The potency, as per API, because it was a previous edition which I looked into, so the potency is given for 2 years, but in the latest notification, it can be kept for 5 years, and indefinitely when prepared with metals and minerals. In case of metals and minerals, the new notification guidelines for 10 years of usage. Then the pharmacological aspect. I've covered 2 points in here: properties and contraindications. Sir has already discussed the various properties. Yes, the mature or the Puran Guggulu, the properties are Laghu, that is, it has a lightness property. Ruksh, dry in nature. Vishad, it has clarity. Sukshma, Sar, mobile. Ati Lekhan, the scraping action it has. And the fresh or Naveen Guggulu, on the other hand, is Snigdh, Pitchil, Bringan, and Brishad. So they are antagonistic in their actions when we use mature and fresh Guggulu. So the mode of action, and again, it's a difficult task for the pharmacies or for the physician to know what kind of Guggulu they are getting. For the pharmacy, if they are procuring fresh Guggulu, they can age it in their own facilities. But when we go to the market, we don't know whether it is prepared with a fresh Guggulu or a Purana Guggulu. So that is again a matter of concern. But in case when we are procuring, we ensure we procure it in advance so that we are particularly aging it for 1 year. Then Karma, Deepan, Balya, Rasayan, sir has already discussed these. Again, this is a verse I have taken from Sushruta Samhita, where he has described Guggulu as a kalp formulation in Vat Vyadhi Chikitsa. I would just like to highlight the Takshanya Oshanyat Kaf Vatagna. Because of its Tikshna and Ushna properties, it alleviates the Vat and the Kaf elements. Saratwan Mal Pittanut. So Malapit elevation is by the Saragun, by the mobility or the mobile activity of Guggulu. It alleviates the Pitta condition. And then Agni Deepanam, it's by its Sukshma Gun, by its minuteness property, it can go to at the level of Dhatu Agni. From Jatharagni to Dhatu Agni to Bhutagni, it can activate. Puti Kosh elevator, because of its Sugandhi Gun, it can curb the bad odors in the body. Now, contraindications, again, Ashtang Sangra Uttarsthan, Ati Matropyukta, any drug which is used in excess may cause certain ailments. And the ailments which have been described are Timir, that is refractive errors of the eye, Shosh, degenerative-like conditions, Kleb, impotency, Karshya, emaciation, Moh, syncope-like conditions, Shathil, that might be because of, if you take it in increased dose, it may cause loose stools. So that may cause weakness, and Raksha, dryness of the body and tissues if we take excessive use of Guggulu. Now, one thing is they have advised, because these conditions can occur if you take it in excess. So it is better to avoid using Guggulu in these conditions unless you are doubly sure about the processing the Guggulu is being done with. Now I come to Yogaraj-guggulu, which has first been described in Chakradat and then Vishajya Ratnavali because it's a compendium. It has taken up this formulation from Chakradat under Amvat Adhikar. Now, sir already discussed these. One thing I would like to highlight here is the third last line. Sammardhi sarpisha gadham snigdhe bhande nidhapir. This is a very important line which should be taken into account when we talk about the Guggulu formulation. Sammardhi sarpisha gadham. It has to be triturated with grit. And snigdhe bhande nidhapir. It has to be kept in a snigdh vessel. So the amount of ghee we are using, that is very crucial when we talk about Yogaraj-guggulu and the pharmaceutical actions, the pharmacological actions we are going to extract out of it. And Yograj iti khyato yogo amrit opamam. So it has the action like amrit, but we have to formulate it as given above. Now these are the various ingredients. Sir has already discussed them. It has chitrak, pipli mool, yavani, both kind of jirak. All of them most of the drugs are Deepan Pachan in this formulation. And then we have Ushnavire and Katuvipak drugs. And then Shuddha Guggulu is added 27 parts. We have 27 herbal ingredients and Shuddha Guggulu is added in 27 parts. And Grit, that is API, mentioned it as 1 part, but in the textbooks, it is not given how much quantity. It's like quantity sufficient that the Vaidya feels. Now, these are the raw materials which we used for the preparation of Yogaraj-guggulu in our pharmacy. This is Ashuddha Guggulu. The Shodhan method employed here was Giloy and Dashmul Quat. And these are the various raw material and herbs, including we have 2 very key ingredients here, Sandav and Yavakshar, which have their own role in the formulation. Now here the preparation of Yogaraj-guggulu. The guggulu is crushed, dissolved in the quat, boiled at mild fire. Temperature should be less than 60 degrees Celsius. On complete dissolution, the guggulu is then filtered through a muslin cloth. Forceful expulsion is required while straining. The filtrate is then again boiled. The temperature is again has to be at a minimum level. And then the filtrate is again boiled till Guggulu Pak Lakshanas appear. Now this is here for Guggulu Shodhan. We have prepared a quath of Giloy and, I've written it Trifla 1 part each. Actually, we have given Dashmul also. So Dashmul can be taken as 1 part. And then 4 times water is added. When we process Guggulu, one thing which is key is the decoction has to be done to half reduction. Because otherwise decoctions are prepared by 1/4 reduction. Like if you're using 4 liters of water, you have to reduce it 1 liter in normal quath preparation. But for Guggulu Shodhan, we have to reduce it only by half to get the desired phytoconstituents in the decoction and to reduce the additives from the herbal products, which may act as adulterants or reduce the ratio of Guggulu in the final compound. So this is the filtration process. This is the expulsion being done. It was around 600 grams of Guggulu, and from that, this is the filtration which we did after boiling the Guggulu in Dashmul and Giloy. Now, Guggulu Pak Lakshan, how do we identify when to stop the heating the Guggulu? Now, cardinal signs during the procedure, the Pak material gets sticky. The features are similar to the Rasakriya or the Avleh formulation, Gud Pak, Avleh and Guggulu Pak. They go on similar lines. When we pour Guggulu into a bowl of water, it should settle down without spreading. So I'll just play the video so we can see. So it immediately settles down at the bottom of the glass of the bowl of water. So that is the cardinal sign or the Pak Lakshan. After this, we let it cool down to say 35-40 degrees, and then we add the Prakshep drugs. Because if we add the Prakshep drugs at higher temperature, the active constituents, they get lost. Now Guggulu Vati, now the next steps, addition of Prakshep drugs is done. Now, when we are adding Prakshep drugs, when we are powdering the herbs, it has to be a very fine powder of the herbs, and Yavakshar and Sandhav Lavan, they are powdered separately. They are not powdered with the herbs, they are powdered separately, then mixed, and then we add it to the this Guggulu Pak. Then addition of Yograj, small quantities are added while adding the prakshep dravya till the prescribed quantity is used. The material turns dough-like, which is easily molded into vartis with hand, or pill cutter equipments can be used to cut and roll 500 milligram vartis. That is the dose we keep. And then the temperature while drying should not exceed 60 degrees. That is what API says, though we do the drying in at room temperature. They are dried and stored in suitable airtight containers for future use. Now let us talk about a very important aspect, that is standardization. And here I have discussed the standardization protocol as described in API part 2. Then we have a study from the CCRS, then the parameters of disintegration time and hardness in case of Guggulu, excipients which can be used, then a traditional pills vis-a-vis the compressed tablets we have in the market. Now API part 2 describes Yogaraj-guggulu as a dark brown spherical vati with a spicy, pleasant odor and a stringent taste. Now, these are the various parameters, but we don't find the parameter of disintegration time or the tablet hardness mentioned in the API. Then this is again a report which was published by CCRS on the pharmacological profile, safety and toxicity of Yogaraj-guggulu. Here that is a little hazy, so I've just mentioned it. The hardness here is given as 8.33. The disintegration time does not disintegrate even after 60 minutes. As Dr. Sabnis already said, if the Guggulu does not disintegrate, how it acts. So what is in practice and what we teach our students is the Guggulu always has to be consumed after breaking it or crushing it. Either if you have good dentition, then you can crush it with your teeth, or otherwise we advise the patient to crush it outside and then take it with the suitable decoctions. But what if the patient compliance gets poor, and then if it does not disintegrate in 60 minutes, it may come out as such. So this is the factors which govern the disintegration time for Guggulu. Now, this is an article which I found, and it's an old study in 1994. The link is here on the right. The processing liquids used for Shodhan procedure, they affect the disintegration time. Here it was found that the Guggulu which was processed in Gomutra, that had a better disintegration time than the other methods of Guggulu Shodhan. Then whether hammering was used for processing or not, that again led to the hardness of the resin if it is hammered. Then amount of prakshep drugs added as powder, that also amounted to the hardness of the tablet. The more the amount of prakshep drugs, lesser the hardness. Then the 1994 study prepared Guggulu tablets with wet granulation method, and they used two excipients in it, the Guggulu powder and talc dugdha pashan they used, and sodium bicarbonate as a disintegrating agent. Talc is not actually a disintegrant, but it promotes the disintegration by increasing the distance between the particles of the material and thus helping in disintegrating them easily. So this was a study where Ayurvedic excipients somehow, because sodium bicarbonate and talc both, they are being used in Ayurveda also, to use them as reducing the disintegration time for Guggulu. This is a very interesting study. Then we have a comparative study on the quality control test. This is in reference with Triphala Guggulu, but I found this very good analysis where all the methods of preparation they are being discussed. The Sompak Vidhi, Suryapak, Analpak Vidhi, and then the direct compression method was used. And here we can see the change in the disintegration test. Sompak Vidhi, it disintegrated in 75 minutes, Suryapak in 64, Analpak in 147 minutes, and direct compression tablet that was disintegrated in 22 minutes. And then the hardness test, we can see the results. So here, and again, one more feature is volatile oil content, which was secure in the Sompak Vidhi, but the other, both Suryapak and Analpak, they lagged it. And then the direct compression, because there was no direct use of heat. So maybe that is the reason the volatile oil content in direct compression method is better.
Gulereji, I'm going to have to do that horrible thing of asking for us to draw your presentation to a close, because we are...
Okay, sir. And this is the latest study where cross-povidone, a super disintegrant, was used to prepare the compressed tablets. Then traditional pills versus compressed pills. Obviously, the hardness, disintegration time, weight variation, they are covered when we use the compressed tablets. But the medicinal properties of grit, they are devoid in the compressed tablets. Now, guglusomes, the concept of guglusomes. Because of the guglusterones, they can be converted into tiny vesicles and maybe, and they are considered as non-toxic and biocompatible. We can look over at the study here. And then again, the enhanced topical drug delivery systems. Here again, I would like to second the thoughts of Dr. Subnis, where Guggulu can produce synergistic effect with modern drugs. So here, Brufen ke saath ye study hua tha. This is a study which was done with Brufen, and paroxicam also was used, then phenylbutazone, where the drugs can interact with, not exactly interact, but they can produce a synergistic effect. So dosage has to be kept in mind. So the indications, sir has already discussed it. The main thing I would like to cater to is it kindles the metabolic fire, helps in increasing the tejas component in the body, helps in alleviating the vata disorder. So when fire and the vata element are taken care of, then you can use the formulation anywhere. Now, Guggulu Sevan Vidhi, when used in vati form, it is advised to use it after crushing the Guggulu pill to avoid delayed disintegration. It is consumed in the Sahitas. It has to be consumed after mixing with Kwat of Trifla, Daru Haridra or Kush, or with Gomutra, Kshar or Ushnodak. And after the digestion, patient is advised to take Yush, Mansaras or milk. Now, this is the Sharangdar Sanhita dose. Ek Shan dose has been advised. The pill has to be of 3 grams. And API advises the dose at 2 to 3 grams per day. This is the main mode of action. I've just covered it concisely, what I feel. There are certain main drug is the Guggulu, certain potentiators, which we have as the Deepan Pachan drugs, the bioenhancing action of the Trikatu and Sandhav and Yavaksha, and the antidote action of Trifla and Grit. Now, I come to the conclusion, Guggulu, because of its Yogvahi and Rasayan properties, is popular as an independent dosage form. The tablet hardness, its disintegration time is a challenge, which can be overcome by use of disintegrants or by patient education if classical pills are being used. Yograj-guggulu, being an alleviator of Vayu and Agni Deepak effect, works in a wide range of disease conditions, hence said rightly to have the effect like Amritam. The concept of Guggulusomes, while opens up new research areas, it also validates the use of Guggulu with medicated decoctions in the Samhitas for internal as well as external use. So with that, thank you. These are the references, the links I have shared, almost all the links I searched for. Thank you for your patient listening.
Praveen, thank you. Praveenji, thank you so much for this very detailed presentation. There's several questions. There's a question in the chat box. How long, what's the duration for use of Guggulu preparations? Number one. Number two, a question that many people ask, this is for yourself and also for Subnisji. Why do we have so many additional additional herbs within preparations? So that's a question. I just want to use that as a start question. So the question, I don't know who would like to take this, Subnisji. You say there are 28 ingredients. Valeria ji, I don't know who would like to address this. Why so many ingredients? Why did our acharyas use this? I want to use that, your answer, as an excuse then to connect with Jerry.
Okay.
So
So,
yeah, Valeria ji, you can answer. You can answer. I'll add on to it.
Yes, sir. You would be the better answer, provide the better answer. But what I think is the 28 ingredients, they balance the formulation because they are working in conjugation. Because most of the drugs, they are Ushna and Katu. Tikta Katuras and Ushna Virya. So we have Ushir also, which is Sheet Virya. And we have Sandhav again, which is Anushna Sheet. So it is a balanced formulation which works at multiple levels. And moreover, we are using Yogaraj-guggulu in a wide array of diseases. So it's basically a clubbed medicine. It's not a single drug which we are using. It's which is single medicine which caters to many diseases. So that is what my take is on this. And Dr. Subnis, you're valuable.
Yeah, there is one principle that is Vriddhi Samane Sarvesham Viparita Viparyaha. So all the ingredients which are of the same quality will enhance the effect of the drug. That is the basic principle coming from Charak Samhita chapter 1, Sutrasthan. And that's very, very important. And second thing, it is not.
So we are losing your We are losing your voice, Mukunji.
So basically, are you getting me?
We are. It's faint,
Yeah.
but
Okay. So basically, all these drugs, they all have synergistic activity. They are all bitter in taste. They are having a collective activity, and that's why the potency of the drug is to be increased to cover the major area of the anabolic disorders. So that's why they have used multiple ingredients, and it has been seen by many researchers that when multiple phytochemicals are used, they have different targets on the organs. And
thank
that
you,
is the
Mukunji.
unique thing. That is the unique thing.
Now, I think this kind of brings out the point you are making about the bitter receptors, etc., etc. Thank you so much, sir. Now, I have please stay with us. The question is to take this idea on to Jerry and to Jerry. The question here is, you can see a logic that is there in the ancient systems. Here is a plant that's been used for thousands of years, 5,000 years, perhaps. Certainly for 2,500 years, documented how it's be purified, how it's be used. Now, there are difficulties here in terms of making it ready for the rest of the world. And you yourself started off by saying there's a challenge. You know, you had a first preparation, it worked, and the second time. How do you bring it all together and kind of make sense? And if we were to take this forward in terms of a large research program between India and North America, that includes both USA and Canada?
Well, I think over here, people are much more linear thinking. When you start with 1,000 variables and you reduce it down to 100 variables, it's still impossible to figure out what's going on. I mean, you have to answer, in my mind, in my simplistic mind, you have to answer one question at a time. For my question initially, why did this simple preparation that I took the first time work, and the same preparation from the same company a few years later not work? I don't know. Maybe the analytical method that Panda used was not correct, and maybe that accounted for it. Maybe it was the same amount of Guggul sterone, but that's a very simple question. There were no other ingredients in this, at least that were on the label. It was just the Guggul sterone. So that simple question, I think, needs to be answered first, and then we can talk about other complexities. But when you start with so many complexities all at the same time, it just seems, I'm not saying it can't be done, but many people just can't accept.
Mukunji, there is a tension.
Yeah. So here we can go for the studies like LCMS, that is liquid chromatography and mass spectroscopy. So such type of studies can explore that why such type of ingredients are to be used. And so as sir has told us, he has asked that why only single variable should be there. And here when it comes to Ayurveda, definitely Ayurveda deals with multiple variables, Dusham, Desham, Balam, Kalam. So there are multiple variables which are to be incorporated. That means the beauty of holistic approach. That's very important. And if we want to bring out the beauty of holistic approach, a single targeted molecule will never give a complete holistic approach. It will lead to certain untoward effects, as we are seeing with statins.
Mukunji, hold on to that thought. I want to see if Baswati ji can add to this, because I feel, Jerry, this tension will remain, because the worldview and the epistemological foundations are vastly different between the Eastern mindset and how we go about doing the reductionist analysis. But it is a healthy tension. It is a healthy tension. And I think a lot of things will come out of this tension if we can manage this tension correctly. Baswati ji.
I don't think this tension is going to be solved until we change the education, because when I was learning pharmacology—
Education where?
The education around the world on how we teach medicine and what's going on in physiology.
Is that an easy one to do?
It could be if you get the right people, but there have to be people that understand You cannot teach people reductionistic organ-based systems and then expect them to put it together when they suddenly get into clinical practice. And you can't expect Ayurvedic people to translate everything when obviously the Sanskrit doesn't translate directly. But when I see Ayurvedic people showing chemistry and they don't understand the context of the audience, that's going to be a big problem, because you're going to have people doing FTIR and LCMS and not understanding what the chemist is thinking about and why it doesn't even work for an Ayurvedic person, but it also doesn't work for a modern medical person. So where are those data left? Like, what do you do with those data? Because
Jerry, have we a perfect mess, or is there a way out of this?
That's a good question. I don't know. I can't answer that.
Perhaps this would be the start of those discussions, and with your guidance, with Baswati there in the States, seniors like Mukundji and Professor Gularia, we can come together, I feel, as a kind of a task force to look at this problem and work on it both ways, on the holistic side with a team in India and the reductionist side with a team across the Western world. I feel that might be. Baswati, would you like to add quickly before I bring Ashleshaji from the Midwest?
It would be nice to have that, but I think what we need is something that's a global online forum where there's interaction. For example, I've been talking with Dr. C.B. Jha on my text as we've been listening. Both of us are here, he's as a participant. And there's a lot of feedback that needs to come from the audience in order for the information that's being presented by the presenters to actually have use to the audience.
It'll be very difficult to do that in 90 minutes. So this is a taster, and I think the taster has generated the interest that we need. I want to bring Ashleshaji before Pallaviji. Pallaviji, you have a question, or I see a raised hand. A question?
Yes. A quick question to the speakers. From the point of collection of Google, when is it termed Navak, the new Google, and the Puranak? Like, what is the time period?
I think Sibnisji is the best person to address this. Please unmute, sir.
So when we get the exudate from the plant in summer seasons, so that is the right time of collection, and from that once we collect it, then till 1 year it remains a Nava Google, and after 1 year it becomes the Purana Google. It is the summer season where we can collect, as in the initial time Baswati ji told that in the moisture we can't collect Google, it will not exert a good results.
Thank you, Ji.
Thank you.
Thank you very much. We are Ashleshaji, your comments on getting us out of this mess. You're based somewhere in the Midwest. Is there some way out of this?
I would not call it as a mess. I would call it only as a complexity from both the sides. I completely agree how Dr. Baswati was saying that yes, it cannot be translated, but at the same time we do have to explain. And I see one positive aspect here, that at least now that we have started teaching Ayurveda very authentically in US, there may be these new students who are coming up, maybe understanding a little more than what we maybe, when I learned back in India, and the focus was a little different. Now with the more globalization or more technology, things are improving, and we would be, I'm positive that something will come out. And the research aspect is extremely, I completely agree with that maybe that kind of initiative can start developing more, opening the more channels and making this more streamlined, or at least we'll get the direction. That's what I could say.
Thank you, Ashleshaji. Satishji, you hold the fort in Canada and with the Canada-India Foundation. Do you see something that we can do here across North America? And that it would also include Mexico. Is there something we can do there out of what is coming out of this very, very rich discussion on a very ancient formulation, and that is revered by all traditions? The Chinese respect this plant and the herbs. the Middle East, people of the Middle East respect this. India, Africa. So is there something here for us from your perspective as chairman of the Canada India Foundation?
No, thank you. I thoroughly enjoyed the discussion and very inspired and excited about the fact that it did work for the first time. I think that's, looking at the glass half full or half empty. I think it did work for the very first time, and that's a very positive and a very, I think, enthusiastic way to look at it. Now, why it did not work? Yes, it's a matter of that how we can run clinical trials on a long-run basis. Like that opens up the chapter. I think the whole discussion which we started for almost a year and a half ago on this subject. I think that's the area we should be working on. And lately, we've been inviting Ministry of Ayush. Actually, we are visiting as Dr. Madan, for our World Health Congress. So our one key objective is to have a meeting with the UHN, University Health Network, where we did the MOU, to have some kind of a pilot project to begin with. So whether that could be, picking up some of these herbal medications on we have heart disease, kidney, diabetes, all those as a part of the pilot project. So picking up some of the key, very focused trials and making sure that all preparation of the herbal medications are standardized, and then building up a database based upon that. And when we have a reasonable, predictable outcome, I think that's the time when we should be pushing it forward more and getting advice from and guidance from Dr. Jerry, Bhaswati ji, and many more. Actually, so I think it's a right platform. We are having that discussion, and we need to keep working on that.
Thank you, Satishji. Thank you so much. Your efforts with the University Health Network, almost all the university hospitals in Toronto, it's a very, very big base from where we can go. And I'm hoping, Jerry, your experience with the National Institutes of Health and the challenges there, and also the kind of interactions that happen between America and India, I feel optimistic that we will be able to work together and develop fantastic programs that will be not only for India and the Americas, but for all of the world. So
I'd like to just mention one friend of mine in Canada that some of you may or may not know, Jacques Bradwin, who is the chairman of psychiatry at Ottawa. He would be very interested, I think, in collaborating with you guys, and certainly the Canadians should know about him and contact him, because he's very good.
Jerry, thank you so much for adding to this, and we look forward to your guidance too as we go on to develop the efforts in Canada, not just in Canada, but for all of North America and the Canada-India collaborations. And as I said, these are tasters. We've got a fantastic discussion going here today, very rich and inputs from all our speakers and very important directions for the future. I leave it here. Thank you for joining us today. Pallavi, all yours.
Thank you, Dr. Madan. So it's been a wonderful, wonderful session today, and seeking blessings from Lord Dhanvantari and thanking Lord Dhanvantari. On behalf of International Ayurvedic League, I thank our special guest, Dr. Jerry Cote and Bhaswati ma'am, for sparing their valuable time with us today. Our expert speakers, the presentations were excellent. Thank you so much, Dr. Mukund, Dr. Praveen. My special thanks to Canada India Foundation. Satish ji, the way you're spreading the essence of Ayurveda is amazing. Thank you. My special thanks to Consulate General of India in Toronto. A big thanks to Canadian College of Ayurveda and Yoga and Tathastu for supporting these webinars technically. Special thanks to our collaborators from Canada, America and Europe, Canadian Ayurvedic Practitioners Association, American Association of Ayurvedic Professionals, thank you very much. Ayurveda Union of Midwest, Ashlesha ji, thanks for being here and gracing the occasion. Thanks to European Ayurveda Association and Association Ayurveda Academy. Very grateful to Y Media and Pravasi Television for supporting us technically, and truly appreciate the efforts of Harish ji and Dr. Madan for make these sessions so interesting. Very grateful to our viewers to be with us today. Our next lecture will be on 11th June and it will be on Rasayan Churn. So with this,